So our next speaker is Dr. Luis Hernandez Miranda. Dr. Hernandez Miranda is a principal investigator at the Institute of Cell and Neurobiology at the University of Medicine in Berlin. Like Muriel, I had the pleasure of meeting and talking to Luis and sharing an Uber ride, all stuffed into one car to save money. We had the pleasure of driving with him to the airport with his lovely student who was also attending the Breathe conference. Luis was a presenter at the Breathe conference. And I have to say that Becky Martini, who is the vice president of the network, and Linda Dokas, who is the co-chair of our research advisory committee, we were very much impressed with Luis, impressed with his recent work, but also impressed with him as a person. And we really, while he is relatively new to the CCHS world, we really think he is going to be a mover and a shaker in the coming years. So we were happy to award Luis and his team a CCHS network research grant in 2024 to continue his research. His research focuses on the identification of transcriptional and developmental programs that safeguard the development of brainstem neurons important to breathing. And when dysregulated, they result in congenital hypoventilations. So today he's going to talk about FOX2B and LBX1. These are both transcription factors and how they are involved in regulating neonatal respiratory physiology. And so with that, I'm going to hand it over to Luis. Thank you for joining us. So you want to share your screen,
Yes.
Luis?
Yes, yes. I don't know if you can see. Just give me 1 second. I'm trying to go to screens, desktop. Okay, give me 1 second.
Share button at the bottom there.
I may have to leave. I'm so sorry. I think I have to install something. If you
Luis, do you want to send it to me and I can, could you send your PowerPoint to me and I'll put it up, I'll share.
Okay, let me try one more time. Now I can see it. Perhaps you will see it now.
Yep, we're good.
Okay.
Yep.
Thank you very much.
So you first put it in presentation mode, yeah.
Yeah. So first of all, many thanks, Melinda, for inviting me, and also I thank the CCH network for granting us the research grant to continue with our investigations. I have to say that my work has been based on the work of many before me, who have done important contributions to understand how neurons are specified in the brainstem. And from this knowledge is that we have profit, and we apply in codes that have been identified by many, and we have also modestly contributed to them. So as Isabella and Muriel have introduced, so CCH has a presentation with respiratory phenotypes as well as peripheral phenotypes. So the respiratory phenotypes include hypoventilation, central apnea, abnormal chemoreflexes to changes in tissue gases, and in very severe cases, it can lead to respiratory arrest. From the peripheral phenotypes, they can affect the neural crest and the neural crest derivatives. So neural crest tumors can be presented together with the respiration phenotypes, as well as the Hirschsprung disease, which are part of the syndrome characteristic of CCH. So as Isabella introduced, FOX2B mutations, heterozygous and de novo dominant FOX2B mutations are the genetic signature of the disease. They can be alanine expansion or non-alanine expansion, being the latter more serious than the former. They tend to produce stronger phenotypes. In recent years, we have identified several mutations in the transcription factor LBX1. They are presented as homozygous, so they are recessive, and they range from frameshift mutation, missense mutations, as well as point mutations. All of these target respiration, but they can have additional phenotypes. The one that we have characterized the most is the frameshift mutation that was reported in a family, and recently we have found a second family with the same mutation. And we are interested in this mutation because it shows a specificity for one single neurotype in the brainstem that we call DB2, and I will go through it in the next slides. So FOX2B is a transcription factor that is expressed and is important for central and peripheral visceral neurons, whereas LBX is better known for its function in specified somatosensory neurons and also several types of brainstem neurons. So it's not surprising that most mutations of FOX2B will cause both respiratory and peripheral phenotypes because it is expressed in both in the central and in the peripheral nervous system, whereas the LBX mutations that we observe, they are restricted to the central nervous system because LBX is not expressed in the periphery. So these two transcription factors are largely expressed in non-overlapping neuron types. So the function is to provide a fate, to impose a fate into neuron types when they are specified, and they mutually repress each other. So in this, sorry for this, in this drawing, I hope you can see my pointer. I show you somatosensory neurons which coalesce into the nucleus trigeminus, in the spinal nucleus trigeminus. These are dependent on LBX. And what you see here in this pinkish coloration here are visceral neurons. So we have the nucleus tractus solitarius, the area postrema, and two visceral motor nucleus, the nucleus vagus and the nucleus ambiguus. So the two factors will repress each other, but if you ablate and remove from the genome one of these factors, very, very pronounced consequences happen. For instance, if you ablate FOX2B, all FOX2B neurons will not be specified, but the neurons do not die. They change the fate. They adopt an LBX fate. As a consequence, LBX neurons increase in the brainstem. And what you would see histologically, for instance, in a FOX2B null-mutant mouse, is an increase in somatosensory neurons of the trigeminal nucleus. The opposite happens if you mutate LBX and eliminate from the genome. Non-LBX will be specified, and the consequence of this is that the nucleus trigeminus disappears completely, whereas the nucleus tractus solitarius, which is viscerosensory, increases in size. So they are actively repressing each other to impose the correct fate to the neurons, either to be viscerosensory in the case of FOX2B or somatosensory in the case of LBX1. Nevertheless, and despite they are expressed in largely non-overlapping neuron types, there is a single exception for this rule, and it's in the specification of a tiny neuron type that is specifically located in the medulla, which we call it DB2. This neuron co-expresses both factors and depends on, and the expression of both factors is important for the correct specification of these cells. The term D stands for dorsal because they are specified in the dorsal brainstem, dorsal meaning towards the back of our bodies, and they are located from a very, very tiny progenitor domain. I should say that the work of many, including us, have shown that there exists at least 13 different progenitor domains in the developing brainstem from which the whole diversity of neurons emerges. One of these progenitor domains is the DB2. So these DB2 neurons emerge from progenitors that first express FOX2B, as you can see here, and as they become specified and differentiate into neurons, they co-express FOX2B and retain the expression of FOX2B during development, okay? Perhaps before our work, the only or the most described DB2 neuron type was the nucleus, retrotrapezoid nucleus, that locates underneath the facial nucleus. And as Muriel has shown, is super important for mediating the hypercarbic response. That is to mediate increases in acidosis in the blood. And when CO2 increases in the blood, is transformed into carbonic acid. This acidifies blood, and these changes are sensed by the RTN, and these neurons in the RTN entrain the pre-Boxinger complex, which is the respiratory rhythm generator, to increase the respiratory output, and therefore allow us to breathe faster. And the consequence of this is that we can eliminate in an increased manner the CO2 from our bodies. Dislocate these cells, as you can see here. These are the retrotrapezoid nucleus neurons. They co-express LBX and FOX2B, and you can see here in yellow. And here you can see them in red because we are illustrating only FOX2B to differentiate them from the motor neurons of the facial motor nucleus. We use CHAT. So CHAT is only in the motor neurons, but not in the retrotrapezoid nucleus. You can observe here also LBX in green is underneath the motor neurons. And here you see the co-expression of both LBX and FOX2B for the RTN. Several years ago, in 2008, the group of Christo Goridis and Jean-François Brunet introduced into the mouse genome one of the most frequent FOX2B alanine expansion, the FOX2B27 alanine, which is a 7 increase expansion. And what they observe is that it behaves like in humans, it's heterozygous dominant. It produces a very severe phenotype with a strong hypoventilation and respiratory arrest at birth. Mice carrying the mutation, the heterozygous mutations, cannot sense CO2 at birth, and they show a specific agenesis of the retrotrapezoid nucleus. A few years later, when we identified the LBX friendship mutation, we introduced into the mouse genome, and we observed a very similar phenotype. So meaning the mice had a very severe hypoventilation. They did not respond to CO2 challenge, as you can observe here. So this is the respiration in normal air, and this will be in hypercarbia. So when we increase CO2 in air, so no response. And since we use antibodies for LBX and FOX2B in our studies, we observe that the mice exclusively lack those neurons. So many LBX-dependent neurons were present and normal, but only those that would co-express FOX2B were lacking in our mice. So not only the RTN, so there were additional neurons affected that co-express LBX and FOX2B. Or in other words, these are the DB2 neurons. Also, years later in 2011, the same group of Brunet, Goridis, and Jorge Gallego show that the specific elimination of the RTN does not cause respiratory arrest at birth. So the mice have a mild hypoventilation during the initial days of life, but as the mice mature, they can reach normal respiration. At birth, mice that do not have RTN cannot sense CO2 at all, but as they mature, there is a gain, and they can still sense a little bit of CO2. So we have reproduced the same results using our LBX friendship mutations. So when we eliminate and restrict the mutation to the progenitor cells that would generate the RTN, we observe exactly the same phenotype. So the animals hypoventilate very mildly at birth, but as they mature, they overcome this, and they can have a normal baseline respiration, and they can still respond to 1/3 of what it would be expected for a control mouse when you expose them to CO2. So our experiments show a very large similarities between the FOX2B alanine mutation and our LBX friendship mutation, but in both cases, the RTN, the elimination of the RTN does not reproduce the severe cases of CCH. And so we were starting to think, since FOX2B and LBX1 were the only neurotype that was eliminated in our LBX friendship mutant, is that we thought that maybe that cell population would be responsible for the whole respiratory phenotype. So one thing that we did was to restrict the mutation to FOX2B expressing cells. So only FOX2B expressing cells would have LBX friendship mutation in them. And what we observe is that they fully recapitulate the phenotype seen in both in the FOX2B ALA constitutive mouse or in our LBX friendship constitutive mouse. So they have severe hypoventilation, they have pronounced central apnea, they did not respond to CO2 at all, and they die within the first hours of life. We observed that, again, that in addition to the agenesis of the RTN, many other neuron types expressing LBX and FOX2B, the DB2 neurons, were lacking in our experiments. So, as I mentioned, LBX is expressed in DB2 neurons, but there are additional neurons that do express LBX, which are also called D. So as I mentioned, D stands for dorsal, B stands for class B, because in the dorsal brainstem, there are two neuron classes that are specified, class A and class B. Class A depend on a transcription factor that is called OLIC3. Class B depend on LBX1. Okay, so all DB neurons express LBX in different combination. Only DB2 co-expresses FOX2B and LBX, okay? So one thing that Kesui and Jilin did in the lab, these are two PhD students, extraordinary and hardworking students who have done most of the work that I will present from now on. What they did is they restricted the LBX friendship mutation to each DB neuron type using different Cree driver lines. But they restrict the mutation to DB1 neurons, we didn't see any phenotype. The animals are perfectly happy. When we restricted DB3 or DB4, the same. There is no respiratory phenotype or nothing that we can detect as abnormal. It's only when we restrict the mutation to DB2 neurons is that we have the pronounced respiratory phenotype seen in the LBX homozygous friendship mutant mouse or in the FOX2B ALA mutation. So that was our initial confirmation that DB2 neurons may be relevant for the congenital hypoventilation phenotype. So why this becomes so specific? So something that we have been doing on the last few years is trying to look at the cooperativity of these two transcription factors. And the very first thing that we ask is if these factors could make heterodimers, if they are physically interacting to each other. So what we observe here in this plot that we have, it comes from a co-immunoprecipitation study, is that the LBX wild type and the FOX2B wild type are capable to make heterodimers, but the friendship does not. It doesn't really like binding to FOX2B. This seems to have a molecular consequence because we have observed, this is a ChIP-seq experiment. We are now doing cut and run and different variations of the proteins. What we see is that the LBX, when it's present with FOX2B, is capable to bind to 801. 801 is an important transcription factor in the development of RTN neurons. And this binding is not present in the LBX friendship mutant protein. And as we have seen before, the consequence of this is that the RTN is never developed in the LBX friendship mutant because we lack this cooperativity. So with these experiments, we started to conclude that the DB2 neuron, the LBX friendship is specific for DB2 neurons, and the DB2 neurons may be responsible completely for the respiratory phenotypes that we observe. And this is something that Kisui and Jilin continue working on, but something that they wanted to do was to know how many DB2 neurons exist, because the DB2 population is small, but only the RTN was known. So something that they wanted to know is how many neuron types exist. So this is one thing that they did first, but also what they wanted to know is if these neurons are cell autonomous sufficient to produce the phenotypes. Okay, so one thing that they did is they used an intersectional strategy to express a dread receptor that when it's activated, it will silence neuron activity, okay? So they express this red receptor in DB2 neurons using two recombinases, FLIPO and CRE, driven by FOX2B and LBX1. So only cells co-expressing these two factors will have the red receptor and will be silenced when we provide the ligand, which is CNO. And what we observe here, you can see the recordings in neonates and in adult mice before CNO, and in CNO, control mice do not change significantly. But mice in which the DB2 neurons are inhibited, they hypoventilate very pronouncedly. Okay? So this was a confirmation that the DB2 neurons cell autonomously regulate respiration and the hypoventilation phenotype. This is caused by a pronounced reduction in tidal volumes, which is the amount of air that is taken by each breath in our bodies. And also what we observed is that the silencing or the inhibition of these cells in neonates completely abolish the CO2 response. But as the animal mature, there is a gain in the response. So animals can sense a little bit of CO2. It's not as efficient as it would do in control animals, as you can observe. It's only 1/3, but still there is a gain in the CO2 sensing by animals in which they are silenced. So as I mentioned, the second thing that they wanted to know is to define, what they wanted to define is how many DB2 neurons exist in the body. So what they did is another intersectional strategy to express the tomato fluorescent protein only in neurons co-expressing LBX and FOX2B, or that have a history of expression of both transcription factors. What we observe here are brains. It is the complete brain, but it has been clear. So all the fat has been removed. So we can see only the fluorescence of the cells that of the DB2 that would co-express tomato. And here we see the histology, just a section with immunofluorescence. In the next slide, I will show you a video of how this looks. So here, all the white dots that you see are DB2 neurons. It's the complete brain, but it's clear, so we only see the fluorescence of the cells mark. We are observing from different views, dorsal ventral. So here we have a magnification. Every of these dots is a neuron. So what you see as a blur, these are axons that are interconnected. This is a low powerful magnification, but we could see a little bit of the connections of the cells. And these are some of the neurons that we have defined. So the intertrigeminal region, the epiphyseal, the RTN that was known before us, vestibular neurons, and a group of caudal populations that are many, and I will show you more details in the next slide. So with this approach, Kesui and Angeline defined 12 different subtypes of the DB2 group, or 12 subgroups. So before our work, the RTN was the best known, and also the intertrigeminal group. Also, they are called peritrigeminal neurons. They locate around the trigeminal motor nucleus. This has been very, very well characterized, much more the RTN than the intertrigeminal. And what Kesui and Jilin found is these additional ones here. So a group in the vestibular nuclei, a group above the facial nucleus that we call epiphyseal, two groups around the nucleus ambiguous that we call epinucleus ambiguous, meaning above, and peri means around. Also a group that is located underneath the trigeminal nucleus that we call infra-trigeminal nucleus, and a group that is around the nucleus tractus solitarius that we call epinucleus tractus solitarius. So many populations, and one thing that Kesui and Angeline wanted to know is if all will play a role in respiration or only subgroups. So to know this, what they did is first map lineage trace each of these subgroups to know from which particular part of the brainstem they are generated. The brainstem in the early development is divided in segments that are called rhombomeres. DB2 neurons emerge from rhombomer 2 to rhombomer 6. Rhombomer 2 generates the intertrigeminal region, but this is the only DB2 neuron type that was not affected in our LBX friendship mutant. So we speculated that maybe only DB2 neurons emerging from romomer 3 to romomer 6 were responsible for the respiratory phenotype. And these are the romomers that we investigated. So to this end, we use 3 different Cre driver lines that are expressed specifically in these romomers. For instance, the HOXB Cre is only expressed in the romomer 4. The EGR2, formerly known as CROX20, is expressed in romomer 3 and romomer 5, and the HOXA3 is expressed in romomer 5 and romomer 6. So using these Cre lines, we restricted the mutation into the LBX friendship mutation into these romomers and analyzed the respiratory phenotypes. So when we restricted the mutation to romomer 4, we observed no phenotypes. The animals breathe normally and respond also normally to hypercarbia. If we restrict the mutation to romomer 3 and 5, what we observe is a very mild hypoventilation that is caused basically by a reduction in the frequency, in the respiratory frequency, but there is no change in tidal volumes. Tidal volumes are the same. These animals fail to respond to CO2. When we restrict the mutation to romomer 5 and 6, we see a more pronounced phenotype that is characterized by very, very long respiratory cycles, meaning that the frequency drops significantly, and also the tidal volumes become shortened. As you can see, they are reduced. Okay. We also see a very pronounced apneic phenotype. So the mice do not breathe for periods of 30 seconds on average, but nevertheless, the animals survive over the time. So if I can summarize, so the elimination of DB2 neurons from romomer 4 caused no phenotype. From romomer 3 and 5, there is a mild reduction in respiratory frequency, and there is an impaired CO2 response. And the elimination of romomer 5 and 6 recapitulates this impairment in CO2, because we are also touching romomer 5, but it produces a more severe respiratory phenotype and respiratory instability. So respiration becomes very, very chaotic. So histologically, on these mice, we observe only the elimination of these 3 groups, but all the caudal populations were not affected from the restriction of our mutation. So that, for us, imply 2 possible scenarios. Option 1 is that these neuron types are not emerging from romomer 3 to romomer 6, or hypothesis 2 was that the Cre line that we used to recombine romomer 6 was not completely recombining this rombomer. So we tested this possibility. So we started, I will skip this one. So we use an additional Cre line that is known to be expressed in rombomer 6, 5, and 6, which is the MAFB. And as you can observe, MAFB recombines completely rombomer 6, but the HOXA3 does not. Okay, so in this schematic view, only MAFB would recombine completely rombomer 6, whereas the HOXA3, only the most caudal part is not recombined. So we use this MAFB to restrict our LBX friendship mutation, and what we observe is a super pronounced phenotype. The animals barely breathe. Unfortunately, they die within the first 2 hours of life, and they lack the caudal population. So here I show you one of these, which is the perinucleus ambiguous. This is the nucleus ambiguous, and the cells, as you can see, are completely gone. So if I could summarize the results by Kesui and Jilin, I could say that our work has defined new neuron types or subgroups in the brainstem of the DB2 lineage. We know that the RTN is essential for the CO2 chemoreflex, whereas the rostral rombomer 6 generate DB2 neurons are important for ventilation and serve as an anti-acneic center, perhaps, and the elimination of caudal DB2 neurons produce a respiratory rest. We still don't know how these cells are interconnected with the respiratory oscillator with the respiratory rhythm generator. This is what at the moment we are trying to investigate. And we also don't know if there is a single population or is the compound loss of the cells what causes the phenotype, okay? But what happens with these cells? The cells do not die. What happens is that LBX1 differentiate overrides the function of FOX2B to impose somatosensory phenotype. As you can see in the control, these are the somatosensory trigeminal nucleus. And in the mutant, I hope that you can see it's very red because now we have here all the DB2 neurons in the mutant mouse. Do you see it? Okay, thank you. It's because here I have just the pictures of everyone and I don't see this part of the presentation. But anyway, I think with this, I would conclude our recent findings. I would like to acknowledge, first of all, Kesui, who is the first author. He's in the audience. Maybe she can say hello. She did a very beautiful work, and she has been working very hard, also complemented by Yilinsha. And also in the audience is Fancy Meng, a new student who is also involved in these experimentations. And also I want to thank very much our funders, and especially the CCH network for the opportunity to continue with this work. And with this, I would finish and I would be ready for questions.
Thank you. Do you want to unshare so we can see faces and I can see who's.
How do I do this? Sorry.
Should be at the bottom, a little green button.
Give me a second. Green button.
Oh no, that I don't know. Where's the stop share?
Yeah, I see.
Okay. Okay, on any hands, I am looking for, thank you so much. Very interesting. James, go ahead.
Hi there, just wondering, based on the research you've done, if there was a therapeutic treatment, for example, on the progesterone basis and research that's been done, which assisted somewhat, would it be more important to get in early, so to speak, and test that early if it was on a patient, based on what you know already, i.e. the older they get, would there be less and less of a response to something which did help the DB2 neurons, so to speak?
Yeah, so I mean, we have not tested any drug yet on our animal models. We are planning of doing this. We are just trying to find what would be the best therapeutic approach to use it. I think the animal models offer a good opportunity to test many things because we see we can dissect different phenotypes within the whole congenital hypoventilation spectrum. But we have not tested yet anything. But we plan.
Go ahead, Linda.
So you seem like the perfect person to ask if there would be a developmental window where you might treat pregnant rats, we'd start with animal studies, to affect the formation and differentiation of the RTN. When does the RTN form developmentally? And would a period just before that be a therapeutic window where you could use SAHA or some other drug?
I think it would be E10.5. From E10 to E12, could be for E10 to 11. Actually, you need only 1 day to specify all the neurons in the mouse. So in the mouse, I think it's a good approach to do it, and I think it would be very interesting to that Muriel and Isabella does the experiment. You only have to treat for 1 day. Also for humans, it's a little bit complicated because since most of the mutation would be FOX2B and de novo, we would never know maybe in advance.
And I can't stop without saying that is such an elegant presentation. And please give my compliments to the people in your lab and
I mean,
you too.
Maybe she can show
her here earlier.
She did all the work.
There she is.
Hi, thank you.
Good to see you again.
Yeah, nice to meet you. Okay.
She was great. She's the student we met. Go ahead, Linda, that you're done, Nathan? Nathan? I know I don't say it the French way.
Yeah, it's both are fine, really. Thank you, Luis, for this great presentation. I had one question Regarding, you know, the neuronal groups that you show are affected when you use the CRE that is specific to Rumbamer 6, which basically identifies all those neurons that are not RTN that might be very relevant to the pathology. Do you know, or do you plan maybe to look at them in the mutant that you mentioned, that Muriel mentioned also, which is the 27 alanine mutant? So basically my question is, because in those mutants we know, as it was mentioned, that basically there's only 20% of the RTN that is remaining, but as far as, and we've discussed this many times, there's not been a huge characterization of the other nucleus that might be involved, just because the RTN was so striking that, you know, it really, it was obvious back then. It's also keeping in mind that was 15 years ago, and what you're showing right now with the 3D, you know, approach of the embryo, I think we've gained a lot. So maybe, is that something that has it been done, or do you guys plan on doing it?
I think your question is very relevant, and I think Muriel wants to do it. I think we will see this from Muriel if I'm not.
Yes, but I need your mice, Luis. Please send them to me.
No, absolutely. No, I think, I mean, you know, the thing is that before our work, and this recent work, the DB2 neuron group was very unknown. Okay, only the RTN and a little bit of the peritrigeminal or intertrigeminal region, done also by Brunet and Huda Sogbi. Independently, they had been working on this group. But apart from these two subgroups of DB2 neurons, the rest were not known. So this is our characterization. What we are doing at the moment is working on the caudal populations and try to know the molecular signature so that we can access to them physiologically, that we can interrogate the function. But first we need to reveal the identities, because all what we know is that they express LBX or FOX2B. We know the location, but more than this, at the moment, we don't know.
Right. Yeah, thanks.
But the CCH network is at the moment funding us to investigate this and try to find it out.
And so all of these FOX2B DB2 neurons, Lewis, like just like in the RTN are gone in CCHS patients, these other groups are gone also.
From the FOX2B ALA, the work by Brunet and Goridis in 2008, they only show the RTN. All other FOX2B neuron population are present, are anatomically present. Everything that is FOX2B dependent is fine, except the RTN. Unfortunately, I don't know if Muriel would share a few FOX2B ALA brains. I can test for LBX neurons, because I would imagine that maybe the cooperation of these two factors is affected in both mouse models. So I would expect in the FOX2B ALA that LBX DB2 neurons, LBX FOX2B neurons are gone, not only the RTN. So what it was investigated at the time was the RTN, because only the RTN was known. But I think now it should be.
No, you're right. I think we should look much more carefully onto the other FOX2B groups in the brainstem. That's for sure.
Yes, but the constitutive, when you have a heterozygous FOX2B ALA, everything FOX2B is normal except the RTN, but it's not known if the DB2 are affected or not.
And I think this is what you said, but I just want to clarify it, because your research is pretty detailed and meticulous. When you are missing like a FOX2B, I think this is what you said, like you have a FOX2B mutation, there's overexpression of the LBX1 in those?
Yes.
And the opposite happens if
the
you're
other
LBX,
yes.
there's an overexpression of the other one.
Yes, both in the central and the peripheral nervous system. So for instance, I show you phenotypes with my drawings in the central nervous system, but if you mutate FOX2B, you would see that the cranial ganglia, which are visceral, they become kind of dorsal root ganglia, so somatosensory.
Any questions? I do have a question in the chat from one of our patients. Is there a possibility, and I think this could go to anyone on the call. Is there a possibility for brain research for CCHS patients who pass away and would be willing to donate their brains, bodies to research, similar to CTE research? Would that be useful?
I think
To
absolutely.
get a CCHS brain from a patient?
Yeah, I think this would be central. I think it's something that many can profit. I mean, from studying these specimens. Yeah. We have not taken any at the moment.
It's hard to identify. I mean, again, for a patient population, the patients had to die. And so that's not a good thing for us, let's say, but a good thing for research for sure. But the other thing is, say that we could set up a program, like this. How would the tissue need to be preserved for you guys to get anything out of it? I think that would be important, don't you think?
I see Muriel raising her hand, and maybe I'll let her talk and then I answer.
Yeah, the point is that so far the RTN has not been identified in human tissue. There has been one paper from a guy from Paris who did it, but it was, to my point of view, not properly done. So far we don't know exactly where is the RTN and whether it exists and whether it is affected in CCHS patients. So it would, of course, be extremely interesting to have tissue from patients, but we are far from being able to map everything and know what is missing, where is where, what is where, and etc. So that's my point of view. So we still have to work on it. Sorry, Luis, I took you.
No, I agree. So for preservation, there are many ways that we can discuss by email. But Muriel is completely right. So still we need to investigate, try to find these respiratory centers in the human.
I don't know. Dan at the Breathe conference I thought Natasha Kumar was doing some of that research and showing RTN in humans, but I could be misspeaking. Does anyone remember, Linda? Do you remember talking to Dr. Kumar? She worked with Doug. I think that's her name. Linda, you're muted. I thought she was doing some more recent studies that was definitively showing RTN in human
Yes,
brains. But
the
I could be
Australian
wrong.
group.
Yeah.
The Australian group is doing work on human tissue. But, when you stop and think about it, if we got tissue from infant who died with CCHS, if the picture is correct, they wouldn't have an RTN anyway, because it wouldn't have, according to Louise's stuff, it would have disappeared back along the developmental pathway.
Yeah, so I mean, one way to approach is first in the healthy tissue that we could try to identify and then correlate with CCH tissue. Yeah, for sure.
I think that's what Natasha is doing, but again, I shouldn't speak for her. But I do think what we're hearing, though, is that we need to do this work first in mice or an animal model and figure out where exactly the neurons are that we need to look for in their I don't want to say phenotype, but the way they are expressing and looking in a normal, and then maybe get human tissue. Is that right?
Yeah.
Questions? Dr. Hernandez. Okay. Any other questions, guys? Gonna move on. Do we want to take a 5-minute break before introducing Dan, taking a bathroom run, getting a cup of coffee for me in California? Okay, so about 5-minute break for everyone, and you can all stay on and still discuss stuff from the first two talks. But I'm gonna run and get some more coffee.